Hello. Welcome. We will start your advanced micro para compre exam review for your compre exam next week. Tama ba next week ang exam nyo?
Next week pa po, Ma'am Jenny.
Okay. Thank you. So we will start our review with what are the things that we will be discussing today. So syempre, this is a compilation of different topics under your MSMT curriculum. So we will tackle some parts of your Bacte, Para, and Virology na topics. So under your Bacteriology discussion, we will have short discussion or an overview of the common agents of foodborne illnesses. Pero we will focus on your Campylobacter. So these three organisms are the most common bacteriologic agents of foodborne illnesses. And most of them are causes of these different na mga gastroenteritis and diseases because of the hygiene. So common ang mga food and waterborne illnesses on countries that are developing. So like Philippines, marami tayong mga foodborne illnesses. So isa na dyan si Campylobacter. So we will start discussing your Campylobacter species. So as I recall, ano nga ba ang basic characteristics ng ating Campylobacter species? So your Campylobacter are motile with single polar flagellum. And they are non-spore former and they are microaerophilic. So nabubuhay sila in about 5 to 10% oxygen content. So their basic biochemical characteristics are that they are oxidase positive and catalase positive. So mayroon silang catalase na enzyme. So yung Campylobacter natin are agents of gastroenteritis and diarrhea in humans. So it is considered the most common bacterial cause of diarrhea or gastroenteritis in even developed countries. For example, United States. So it somehow surpasses also Salmonella in terms of incidence in the US according to CDC. So there are a lot of individuals that contract Campylobacter annually as a byproduct of being um, contaminated through yung poultry or mga animal products. So when there are direct contact with contaminated na animal feces, pwedeng through contact with another animal, so there can be infection of Campylobacter among farm animals, birds, cows, domestic pets, and mga other fowl flora. So bakit pala siya important? So what if it's rampant among animals? We are human organisms naman. So when there are a lot of bacterial disease burden sa animals, especially on poultry, syempre, di ba yun yung major livestock na kinoconsume natin. And if there are cases na hindi kayang ma-eradicate ang um, organism through cooking or through other methods, it can be passed on from animals to humans. And the humans can also contract the disease. And there are a lot of organisms that chicken flocks contain, and it can spread between different species like other birds, through infected species, and through contaminated water source. So if there are slaughtering na mangyayari, the organisms can be passed from the intestines of that animal product and be passed on to human organisms. That's why it is an important agent of foodborne organisms. So that is your Campylobacter. Campylobacter species. So ano ba yung usual specimen flow natin when we try to investigate Campylobacter? So yung clinical specimen natin will usually follow these patterns. So i-inoculate natin siya sa blood agar and anaerobic blood agar and yung specialized culture media natin, yung CampyBA or CCDA. So, yung mga Campylobacter agar-based blood-free. So, we will culture or plate the the specimen in the following media. So, ano bang ina-expect natin na growth of our Campylobacter on blood agar and whether aerobic or anaerobic. So, we will um, check if there is the presence of translucent moist colonies if blood agar. Tapos if CampyBA, gray to white naman na moist colonies. So what will we do? Kasi hindi naman all the time present itong CCDA or CampyBA in all laboratories. So if nagtumubo na siya, we will of course proceed with the basic step. We will do gram stain and then we will observe for curved gram-negative rods. Kasi yun, di ba, yung basic morphology of our Campylobacter species. But the concern is, it can look like also your Helicobacter species. So what did we say kanina? Your Campylobacter are oxidase positive and catalase positive. So we can do oxidase testing. So after your oxidase testing, we will check if positive ba siya or negative. So if negative, try natin restrict to blood agar and we check after 24 hours. If negative pa din, we will discard the culture medium. But if the oxidase is already positive, so what can we do? We will proceed with catalase testing. And if positive siya, so we can check for other specific products like urease. And then kung positive siya, then it is most probably Helicobacter. And if negative naman, another example of a curved gram-negative rod. So this is just an example of identification scheme. So when we do testing for your Campylobacter species, what are the specific specimen collection or transport requirement natin for clinical specimen? So when we do that testing, we don't have actually special requirement for your detection of Campylobacters. However, what are the two most common clinical specimen that we use na ginagamit natin to submit sa laboratory? So, we use feces and blood. So, these are the two most common clinical specimen that we use to check for or detect Campylobacter species. So what can we use for those patients na nahihirapan tayong mag-obtain ng stool? So we can do rectal swab for infants or for younger children, yung mga 1 to 2 years old, if ever they cannot provide acceptable fecal specimen. So what are the specimen preservation consideration that we need to take note when we are trying to detect the presence of Campylobacter. So there should be no more than 2 hours ang parang preservation time ng stool, which is generally the case for most na stool specimen. So if there will be a delay of more than 2 hours, so we have to now place that clinical specimen, in a transport medium. So we have several examples of transport medium that we can use for your preservation of stool sample. Example na lang yan, yung fecal enteric plus, and yung isa, yung more common, which is your Kerry Blair. So, we will store your stool specimen in 4 degrees Celsius if hindi pa natin siya mapaprocess. So, yan yung isa sa mga few clinical considerations when we are trying to detect for Campylobacter in stool specimen. So, ano ba ang itsura ni Campylobacter on gram staining? So, the gram staining Campylobacter jejuni, if we will be using it from a primary isolation plate, is gram-negative curved rods. So, we can also do direct gram staining of stool obtained from patients that we are suspecting of Campylobacter infection. So dito, these are just example of specimen na nakadetect tayo ng Campylobacter. So ito, helical or curved ang gram-negative rods. And this is another image of microscopic na examination of Campylobacter species. So after examining your gram-negative rods, we can say na there is a difficulty in properly visualizing your gram-negative na mga Campylobacter species or your Campylobacter species kasi they can sometimes not react under your common na secondary stain, which is your safranin, during the gram stain procedure. So if we have difficulty in assessing if ano ba talagang itsura nito na specimen, you can use carbol-fuchsin or a 0.1 aqueous basic fuchsin to improve the visualization of your smears of stools or those na smear from primary culture. So, that is one way of troubleshooting if we have problems in visualization ng gram-stained specimen ng stool when we are trying to detect Campylobacter. So, another test din that we can use in detecting infection is yung itetest natin if there are fecal leukocytes. Before, when they have lesser abilities or lesser testing methods in checking for bacterial infection or Campylobacter, they do fecal leukocyte testing. However, if the fecal leukocyte test is negative, it does not necessarily mean na wala tayong infection. So it's just used as possible screening test. Pero if malaki yung index of suspicion na baka may Campylobacter infection ang patient, we can proceed to other more sophisticated and more sensitive na testing methods. So ano pa ba ang other way of detection of your Campylobacter species. So we can do um, CIDT. So your CIDT, these are the culture independent tests in direct detection of your Campylobacter in stool specimens. So this is an antigen detection system, your CIDT. So what we do is that there will be an enzyme immunoassay that will detect the antigens in the stool if present ba siya even after several days as long as the stool sample is preserved well. So it will detect for some Campylobacter antigens and it will aid in the diagnosis of Campylobacter. However, is it very useful? Kasi kung very useful, bakit hindi natin masyado naririnig? Mababa kasi ang specificity and positive predictive value of this antigen test. So, we cannot use this as a standalone method of diagnosing Campylobacter species. So what is another method of trying to detect your Campylobacter species? So since we are at in the advent of molecular biology testing, so we can do nucleic acid test or nucleic acid detection. So your nucleic acid amplification testing, it will provide an alternative to culture methods in detecting Campylobacter. So minsan mahirap kasing magpatubo ng organism and maraming ways of being contaminated yung ating culture media. So we can use detection of mga DNA fragments of these organism and we check them if they are present through nucleic acid amplification testing. So meron tayong several nucleic acid-based test methods that we can check and we can use in testing for Campylobacter species. So ginagamit natin ang CIDT sometimes together with NAD or NAD alone in detecting Campylobacter gastroenteritis. Pero kailangan yung laboratory is capable of proceeding with molecular methods or nucleic acid methods in order to help in the prevention of outbreaks. So that is your Campylobacter species. So ano ba ang other, since hindi naman lahat kayang mag-undergo ng testing using molecular methods. So, meron pa ring nagre-resort to the conventional na microbiologic methods. So, what are the culture media that we can use in testing for Campylobacter species? So, pwede tayong gumamit nung sinabi natin kanina, Campi BA. So, this is an enriched blood agar to isolate Campylobacter jejuni. So same lang siya sa base ng Brucella agar and it contains vancomycin, it contains trimethoprim, it contains polymyxin B, and amphotericin as your other na antifungals. So these are some of the antimicrobials na composition ng ating Campi BA. So aside from your Campi BA, we can also use CCDA. So this is an agar base na blood-free. So it is a modified agar that contains charcoal, sodium pyruvate, and ferrosulfate instead of the blood. So these are your selective culture medium for Campylobacter isolation. So most of the time, ang ginagamit natin na clinical specimen are stool because there are successful isolations in using stool samples as your clinical specimen. But not purely for diagnostic. Dapat meron din tayong another set of culture. Pwede tayong mag-perform ng blood culture as an addition to the stool culture to check for actual isolation of Campylobacter or to have a higher recovery rate. So these are your example ng culture media na pwede natin gamitin selectively for Campylobacter. So, ano ang incubation consideration natin when we are trying to isolate Campylobacter species? So, we have to incubate one specimen at 42 degrees and one specimen at 37 degrees, pero with high hydrogen content. And the two sets of these selective plates will allow for extended incubation and higher recovery rate of your Campylobacter. So, pagka daw ini-extend natin yung incubation with this, mas nagkakaroon ng higher recovery rates for Campylobacter species. So, one of the most common Campylobacter species that we isolate are your Campylobacter jejuni. So, aside pa talaga sa culture media, kasi hindi naman tayo natatapos dyan na after we allow the growth of your Campylobacter, we also have to check for the antimicrobial susceptibility like most organisms. So, one of the common diagnostic methods in checking for growth and antimicrobial susceptibility of your Campylobacter is the matrix assisted laser desorption ionization time of flight mass spectrometry, also known as Malditov MS. Sorry. So yung Malditov MS, it is a method wherein we will check using a small amount of bacterial sample mixed in a certain matrix solution, and there will be ionization that will happen in the solution, and it will enhance the detection process. So there will be compounds that will be added in the clinical specimen. For example, HCCA or DHB. So yung HCCA, that is a hydroxycinnamic acid, and your DHB is dihydroxybenzoic acid. So we add those solution in order to allow drying of the bacterial specimen. So after ionization, during an ionization, there will be drying of the clinical sample or the bacterial sample, and it will now be submitted to a laser beam. So when the laser beam is focused on the specimen, the matrix molecules will vaporize and it will carry the organism with them to the gas phase. And during this process, yung bacterial cells will be ionized and they will be charged. So when they enter the electric field, kaya tayo tinatawag na time of flight mass analysis or analyzer, it will measure the certain time it take for the ions to travel a certain distance. And it will compare that detail to a certain database which can detect kung anon ba yung mga certain organisms with the same reference mass spectrum or spectra. So we are detecting organisms down to the molecular mass level. So once na na-compare na natin yung specimen to a certain reference spectrum and there is an increased confidence level na same or similar talaga sila ng identity, so we can say na ito nga Campylobacter nga yung organism present because it is identifiable with the spectrum or reference spectrum na nandoon sa database. So that is one of the things that we can use your MALDI-TOF for detecting certain organisms. And check din natin kung meron ba silang certain resistance to an antibiotic. So, pwede rin tayong gumamit ng MALDI-TOF MS for checking ng antibiotic susceptibility testing. So, yan ang isa sa mga common breakthroughs in identifying organisms. So, ano ba ang clinical impact of the burden ng Campylobacter? So, first, let's take note, ano ang reservoir or saan ba nasa-store or ang habitat ng Campylobacter. So, si Campylobacter can be found on uncooked poultry and wild and domestic animals. So, paano ang transmission ng Campylobacter? Just like most foodborne illness, if not all, syempre fecal-oral route. So, as we mentioned last few slides, the Campylobacter species are the causative agent of several gastrointestinal or extraintestinal infection. So, aside from that, your Campylobacter can cause myocarditis, meningitis, abortion, and other visceral organ inflammatory reaction. But the most common is your gastroenteritis. So, ito yung common na mga condition, and these are the common clinical syndromes associated with Campylobacter. So, your Campylobacter causes a self-limiting gastroenteritis, and most of the time, it will not require antibiotic therapy. So, ano pa yung isang important causative agent of gastroenteritis that should not be given with antibiotic therapy unless stated? That is your enterohemorrhagic E. coli. Most of the time, pagka may infectious diarrhea, nagbibigay ng antibiotic treatment. Pero it should not be the case for all infection. Kasi if we will initiate antibiotic therapy on these gastroenteritis na mga conditions, there can be a sequela or a complication due to reactivity on those antimicrobials. So, kung kay EHEC or enterohemorrhagic E. coli, pwede siyang magkaroon ng hemolytic uremic syndrome if initiated ang antibiotic therapy na hindi warranted. Ano naman yung other complications in Campylobacter species that are not necessarily due to antimicrobial therapy initiation? So, post-infection complication of your Campylobacter is yung Guillain-Barré syndrome, and also reactive arthritis. So, ano ba yung Guillain-Barré or Guillain-Barré syndrome? So, that is a phase of acute demyelination. So, it means that the myelin sheath of some peripheral nerves are being removed. And when that happens, nervous or stimulus cannot properly transfer from one neuron to another. So, nagkakaroon ng delay in transmitting information. And when that happens, there are a lot of neurologic symptoms that can occur to a patient as a post infection complication of Campylobacter species. So, lahat ba ng nagkakaroon ng Campylobacter gastroenteritis automatically will have Guillain-Barré syndrome? Not necessarily. There are studies that indicate na about mga 20 to 30% of patients with Guillain-Barré syndrome have neurologic symptoms 1 to 3 weeks prior sa um, resolution of the gastroenteritis. So, somehow you can have a prognostic indicator or predictive factor on magkaka-Guillain-Barré syndrome ba yung patient or not. Next is reactive arthritis or what we call as Reiter syndrome. And these also follow Campylobacter infection. So remember that the Campylobacter jejuni is the most common bacterial enteric pathogen in developed countries and is an important cause of travelers' diarrhea. So, kailan nga natin to nakita? Pag nagkakaroon tayo ng intake of improperly cooked meat or pwede rin unpasteurized milk or contaminated water. So this is an important bacterial causative agent of food poisoning. That is your Campylobacter jejuni. So why are we studying the effects of Campylobacter jejuni? How does a patient feel or how does a gastroenteritis from Campylobacter jejuni manifest in patients? So, yung Campylobacter jejuni is motile and it will stimulate immunopathological responses within the intestinal tract. And yung motile natin na si jejuni will circumvent the mucous layer of the intestinal epithelial layer. It will interact with the mucosal layer and the lamina propria cells. And when that happens, it will result in recruitment of some dendritic cells, neutrophils, and macrophages. So pagka nagkaroon tayo ng interaction between the organism and the effector cells of our immune system, there can be interaction of the innate immune cells. So, yung sabi natin kanina na dendritic cells, macrophages, and neutrophils, it will interact with the lipo-oligosaccharide present in the Campylobacter jejuni. So, when that happens, WBC and lipo-oligosaccharide, so it will create a massive inflammatory or pro-inflammatory na cytokine response. So, nagkakaroon ng cytokine storm. Because or through TLR or toll-like receptor 4 signaling, and it will induce apoptosis, it will induce ulceration, it will induce malabsorption and barrier defects in the intestinal epithelial cells. So when that happens, ano ang product? Nagkakaroon tayo ng dysentery. So there will be bloody diarrhea. So paano umabot sa dysentery yung gastroenteritis ni Campylobacter jejuni? Nasira niya ang mga host barrier defenses because the Campylobacter jejuni was able to adhere sa apical surfaces of the intestinal epithelial cells, crossing the mucosa and the lamina propria. So it will start with mucosal invasion. So after mucosal invasion, it will stimulate interaction with host effector cells. So there will be hyperactivation of some WBCs, and that will cause release of several cytokine response through the TLR4 signaling. And that will cause disruption, ulceration, barrier defect, and will lead to bloody diarrhea. So sometimes, depending on the Campylobacter strain present and also the immune status of certain patients, it can present with just acute, it can present as a highly acute and severe symptom na pwedeng watery diarrhea without fever or severe abdominal cramps, leading to purulent, bloody, inflammatory diarrhea. So that is the overview of the pathogenesis of your Campylobacter gastroenteritis. So sabi natin, no matter how bloody or how intense the diarrhea happened, it is usually self-limiting and can last for several days to 2 weeks. Pero, in some minority of cases, after the gastroenteritis, there will be the Guillain-Barré syndrome, or pwede rin magkaroon ng reactive arthritis. So there were studies that revealed that the severity of the campylobacteriosis or the gastroenteritis is somehow directly involved in the development of the post-infection sequela of the Campylobacter. So the more nagrabe ang symptoms, the higher ang chance na magkakaroon siya ng GBS after the gastroenteritis na phase. And that is mostly associated with the presence of the oligosaccharide structure in the outer cell membrane of your Campylobacter jejuni. So that is somehow an important observation because kahit papano there can be a certain prediction if magkakaroon ba ng Guillain-Barré syndrome ang isang patient after Campylobacter species or not. So, paano ba jud ang pathogenesis ng Campylobacter infection? It's not generally defined yet. It is not fully understood. Pero alam lang na nagkakaroon ng adherence, toxin production, and then invasion ang Campylobacter, and the cytotoxins that is released in the process will cause the epithelial damage. So nagkakaroon ng cholera-like enterotoxin being released by some isolates, and it will lead to bloody diarrhea. And yung bloody diarrhea generally associated directly with the invasion of the Campylobacter strain. So that's what happens in Campylobacter gastroenteritis. Um, yung sinabi nating reactive arthritis as a post-infection of your Campylobacter, it was noted to be present among cases na merong presented HLA-B27 ang patient. So, a lot of extraintestinal complications like flaccid paralysis caused by immunologically mediated inflammatory response of the peripheral nerves happen, but not related to the presence of HLA. Pero yung reactive arthritis, noted yun na nangyayari among patients with HLA-B27. So ano ba ang clinical features ng ating Campylobacterosis or Campylobacter gastroenteritis? There will be watery diarrhea or bloody diarrhea after an influenza-like na prodromal symptoms, and the incubation period is usually up to 8 days. So again, antibiotic therapy is generally not required, pero kailangan ang conservative or symptomatic management like hydration, ganon. So this is an illustration showing triggering of the humoral and autoimmune response of your Campylobacter jejuni in the development of the Guillain-Barré syndrome. So si Campylobacter jejuni will trigger immune response, and because of that, it will lead to antibody production. And the cross-reactive antibody that are supposedly for Campylobacter jejuni can also destroy some parts of the surface of the ganglioside, which are the lipid A and the ceramide. So yung tinatawag natin na anti-GM1 and the anti-GD1A. So these are the antibodies that cross-react with gangliosides and cause destruction. And these gangliosides are important in nerve conduction. So yung anti-GM1 and anti-GD1A, so these are antibodies that will bind to these parts of the ganglioside, lipid A and ceramide, and it will cause complement activation. And the complement activation will be followed by the formation of a membrane attack complex, and it will lead to disappearance of a voltage-gated sodium channel here, and it can cause detachment of the myelin, and it will cause problem with nerve conduction. So, hindi makaproceed properly ang impulse because of the damage in the myelin sheath. So, which part of your Campylobacter jejuni can trigger production of cross-reactive antibodies? So, yung pading lipo-oligosaccharides. So, that will further cause macrophage invasion, and it can also enter periaxonal space, and can cause scavenging of injured axons. So these mechanisms of immunologic mechanisms can result to the development of Guillain-Barré syndrome because of Campylobacter jejuni infection. So ano yung mga So ano yung mga mechanisms of um, development of immunologic sequela of your Campylobacter infection? So pwedeng molecular mimicry, yung sinabi natin kanina that the cross-reactive antibodies will act and bind to some structures in the gangliosides. Pwede rin na nagkakaroon ng immune responses function, and pwede rin nagre-release ng bacterial toxin ang ating Campylobacter, which can directly damage nerve cells and pwedeng magka-produce ng nerve dysfunction even without the binding of the antibodies. So those are several mechanisms that can lead to the development of Guillain-Barré syndrome because of Campylobacter infection. So even though antimicrobial therapy is not warranted in treatment of your Campylobacter jejuni, it can be useful in the process of treating some other infection caused by Campylobacter. And with that, since sabi natin, nakikita ito sa poultry, so yung mga animals and mga livestock natin, binibigyan niya ng antibiotic sometimes ng mga nagbe-breed. And because of those antibiotic therapy na binibigay sa mga animals, natututo sila na mag-develop ng resistance against those common antibiotics. And there are several studies that started checking kung ano na ba ang percentage or prevalence of resistant strains of Campylobacter species found in chickens. So, ganun. So, ano ba ang resistance of mechanisms ng Campylobacter? So, the four major resistance mechanism of your Campylobacter jejuni are as follows. Pwedeng modification of the antibiotics target or alteration sa expression ng receptor of the antibiotic. Pwede rin na it will not allow the antibiotic to reach its target. Pwede rin na may efflux of the antibiotics, or pwede rin na modification or inactivation of the antibiotic itself through enzymes. So your Campylobacter species can develop resistance to different types of antibiotics through different mechanisms. So when an organism develops this mechanism, so example, si Campylobacter jejuni can alter the structure or function of the target site na usually binabind ng antibiotic in the bacterial cells. So for example, the Campylobacter can modify the 23S RNA found in the ribosomes. As part of the ribosome, and when that happens, hindi siya nababindan ng common antibiotics that target that. So example, your macrolides. So the macrolides are reduced sa kanilang binding affinity, so making them less effective. So Campylobacters can develop macrolide resistance. Example ng macrolide, yung erythromycin. So, if the target site 23S is altered, so there will be a problem in the development of proper antibiotic treatment. So there are several point mutations that can cause problem with the 23S part of your ribosomal RNA. Another is pag ang Campylobacter becomes resistant to common quinolones or fluoroquinolones. So the Campylobacter can acquire mutations in the genes that encode DNA gyrase and topoisomerase 4. And when that happens, itong target ng mga quinolones na DNA gyrase and topoisomerase 4, for example, si profloxacin, hindi na siya makabind properly to those antibiotic targets, so there will be diminishing of the ability to interfere with the bacterial DNA replication and repair. Next, if an antibiotic has decreased ability to reach the target, so the Campylobacter can also develop resistance by changing the bacterial outer membrane, making them more impermeable to certain antibiotics. So, during this time, nag-overlap yung mechanism of development of efflux pumps, which will actively pump out the antibiotics in order to prevent them from entering the cell. So, yung outer membrane changes that happen, it can be parang nalilesen ang penetrating capability of the antibiotic to the cell membrane of your Campylobacter. And as one aspect of that is the altered lipopolysaccharide structure. So yung lipopolysaccharide is an essential component of the outer membrane, and when it is modified, so there will be hindrance in the passage of your certain antibiotics across the outer membrane towards the organism. And one example of an efflux pump present in Campylobacter is the CME ABC. So this efflux pump is associated with the resistance to certain types of antibiotics, for example, quinolones and tetracyclines. And lastly, the presence of enzyme, for example, beta-lactamases or aminoglycoside modifying enzymes, so they can degrade or modify the antibiotics, rendering them non-effective already. So those are some of the mechanisms na present in Campylobacter species, making it resistant to several types of antibiotic. So that is your Campylobacter species as part of your bacteriology review for